Hello, everyone, and welcome to today's Keynote webinar titled Toward Universal Coronavirus Vaccines from Concept to Clinic. This webinar is part of the Coronavirus Virtual Event Series, the 7th in the series. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. I now present today's speaker, Dr. Kayvon Modjarrad. Dr. Modjarrad is the founding director of the Emerging Infectious Disease Branch at the Walter Reed Army Institute of Research and Associate Professor of Medicine at the Uniformed Service University. Dr. Modjarrad leads the U.S. Army's development of a vaccine against COVID-19, co-leads all of the WRAIR's COVID-19 response efforts, and serves as a member of Operation Warp Speed. Dr. Modjarrad is also an advisory member of the World Health Organization's Strategic Advisory Group of Experts, Immunization Working Group on COVID-19 Vaccines and Vaccination. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Modjarrad, welcome. You may now begin your presentation.
Thanks very much, Susie, for that kind introduction, and thank you to LabRoots and the organizers for giving me the opportunity to speak to you today. As Susie mentioned, I'll be talking about a broadly active approach towards coronaviruses, but I've narrowed it a little bit to a subset of coronavirus, the SARS-like viruses or sarbecoviruses. And I'll talk about how we took an approach early on from the first day that the sequences were published of this novel coronavirus towards the development of a broadly immunogenic nanoparticle platform that really anticipates and provides protection against the evolving strains and emerging species of coronaviruses. This is just to say that these opinions that are expressed in this presentation are that of my own, that they do not necessarily represent those of my employer, which is the Department of Defense, and that I don't have any relevant conflicts or disclosures to report. I'd like to start with acknowledging the contributors and collaborators to the work that I'll be showing you today. It takes a lot of folks to do research. It takes a lot of folks to do vaccine development. First and foremost, my own program and all the folks in my team, and particularly Dr. Gordon Joyce, who's my co-inventor on the vaccine that I'll be talking about today, as well as a number of collaborators within our institute, the Walter Reed Army Institute of Research, others across the U.S. federal government, as well as collaborators in academia and industry. And this is a picture of just a subset of our group on a nice summer day in the middle of the pandemic when we were vaccinated the first time around and could get outside and take off some masks for a picture, but we're a much larger group than this now. But we really started out sort of as a kind of a little garage band working on the development of this vaccine. And one of our folks, the third person from the left, one of our scientists in the lab, she's an illustrator, and so she's been documenting the many adventures of our vaccine in the context of some animations that hopefully will make a graphic novel into. So I thought it would be appropriate to start out with a quote, and a quote that reflects the tendency of the public health and the scientific communities after or as an epidemic or pandemic is waning. I've been involved with a number of public health emergencies over the past 10 years. MERS coronavirus, Zika virus, H1N1, and Ebola. And each time there's this introspection that takes place in which we ask ourselves how we can do better the next time around. And it tends to veer into the direction of trying to predict what happens next, rather than trying to take stock in the gaps that we had and shore up those gaps for the next pandemic. And so one of my colleagues, who is really one of the fathers of emerging infectious diseases, was the founder of something called ProMed, which provides a lot of very important surveillance information. Was one of the first that reported this new strange respiratory disease in southern China in December, and his name is Stephen Morrison. He made me aware of a quote that I was previously unaware of, and I'm a big sci-fi guy. I love Ray Bradbury, but I was not aware of this Ray Bradbury quote from 1982, in which he said, "People ask me to predict the future when all I want to do is prevent it." And I think that captures where I want to go with this presentation, what I've tried to imbue my program with, the kind of philosophy that guides the kind of products that we develop. Broad platforms for vaccines, therapeutics, and diagnostics that are anticipating the next potential epidemics and pandemics. So if we go back to 2014, 2016, when there was a huge outbreak, the largest outbreak at that time of Ebola in West Africa, what we saw was that Ebola had been discovered in 1976, and over the course of the next 40 years, essentially, there were multiple other outbreaks in Central Africa, and they were relatively small, relatively self-limited. They burned themselves out. They were devastating to the communities they affected, but they were relatively contained. And I think it was always a sort of a failure of imagination on the part of the research and public health communities that this could potentially blow up in a much denser, more populated region. And that's exactly what happened in West Africa in 2014. And so we were able to get a vaccine for Ebola, but not until after 11,000, 12,000 individuals had perished. And the really difficult thing to deal with is that this vaccine, first vaccine that was licensed for Ebola, had been on the shelves for about 15 years, had been tested in multiple animal models, but no one really felt there was a need to invest in a clinical trial. And so when this huge outbreak occurred, we were already behind the curve, and literally behind the epidemic curve. We were able to get some efficacy data, and this is a lot of the work that I did when I was at the WHO for a year in 2015, in the context of modified clinical trials. But had we had some of that clinical information earlier, we could have really deployed this vaccine almost immediately in the context of the outbreak. So that's, I think, a lesson that needs to be made in terms of doing as much as we can before the outbreaks occur. And even in the context of the current pandemic, we had been working, when I was at the National Institutes of Health, on coronaviruses for about 8 years prior. And we were lucky that we were working on that, because that led to a lot of the quick and rapid development of some of these vaccines for COVID-19, but again, for the millions that perished, the tens of millions or hundreds of millions who were infected before the vaccines were available, and in many ways, in a lot of places, they're still not fully accessible. Despite them being developed and authorized within a year, that still was too late for too many. And some people are not always swayed by the argument of public health and saving lives. So we also try to make the argument in terms of financial impact and economic costs as well. If we look at the center of the screen, MERS, Middle East Respiratory Syndrome coronavirus, which is one of those coronaviruses that is also highly fatal, probably the most fatal of the coronaviruses that we know, but does not transmit well, fortunately. Despite that, it infected infected only 186 individuals in South Korea in 2015. It killed about 20% of those, and yet it caused a $9 billion economic loss for the South Korean economy, contracted the economy that year by about 2%, just from 186 cases. And so, we had been trying to make these sorts of arguments before the pandemic, and we thought another pandemic or another epidemic could cause billions of dollars. Well, the estimated impact is probably going to be upwards of $80 trillion when this pandemic is finally over. And so, what should we do to prepare? There have been a lot of initiatives, particularly since the Ebola outbreak of West Africa in 2014 to 16, to focus on developing vaccines for those pathogens that may likely cause pandemics or huge epidemics in the future. And there's all these different lists that have been developed. And that's one of the areas that I was talking about in terms of trying to predict what's coming next. And so if you look at, and I'll show this in a couple of slides, but if you look at how the pandemics or emerging infectious diseases have progressed over the past century or so, there's a pattern that emerges. There is a, can you see my slides, by the way? Okay, I'm just assuming you can't see my slides. So, I'm sorry, there was a change here. So, I can't see if it's, I have a preview. Okay, there we go. So, here, if we're focusing on the few that may likely cause pandemics in the future, which ones do we focus on? Well, we could go back to the ones that have caused outbreaks in the past and say, well, let's focus on those. But I think we need to focus, it doesn't really, we're sort of fighting the wars of the past rather than fighting the wars of the future. And this comes down to, I think, a basic concept that we keep seeing the same things over and over again, but it feels like we're seeing them for the first time. And what I mean by that is that if you go just to coronaviruses, we had SARS in 2002, MERS in 2012. It was very likely that we were going to see another highly pathogenic coronavirus, because in the interim between SARS and MERS, there were two other human coronaviruses that were identified that caused much milder illness, more of a common cold, but there was an accelerating emergence or accelerating discovery of these coronaviruses. And it shouldn't have been surprising that another coronavirus emerged. Now, the scope and the extent to which it affected global populations was a bit of a surprise, but the fact that we saw another one was really not much of a surprise at all. So here's my point. If you look at the number of viruses of relevance to human populations and human disease that have been discovered since, let's say, the last pandemic, the influenza pandemic of 1918, we see that there's this exponential rise in their discovery. And the slide above is borrowed from Dr. Fauci, in which he put these emerging and re-emerging pathogens on this map every year that he's been director of the National Institute of Allergy and Infectious Diseases. And it becomes overwhelming. There's no way to really address all of these, because if you try to do that, you're really addressing none of them. So the next curve is, I think, a way that we can get our heads wrapped around this, that those virus species group into about 24 virus families that are of particular relevance to human disease. And so if we see MERS, SARS, SARS-2, and our future coronavirus, they're all within the virus family. They're cousins of each other. They have common features, common traits that we can focus on, common vulnerabilities, so that we're not developing a vaccine for coronavirus X or coronavirus Y. We're developing a vaccine for the coronavirus family. And we can take that approach and apply it to different virus families, just as we would for the coronavirus family. And so coronaviruses have been a growing family for us as humans as we've learned about them. And the real major concern has been beta coronaviruses, and that's where SARS-1, SARS-2, and MERS come from. They come from this one genus within the coronavirus family. So there are different approaches to making vaccines, and we've seen the whole extent of this over the past 2 years of some old platforms and new approaches. And I just wanted to first provide this slide to provide some context into which category our vaccine falls into. And it's on the right side is a recombinant protein vaccine. And that is a little bit different than some of the others out there because it's a nanoparticle approach, but ultimately it's a protein that is coupled with something called an adjuvant, which is sort of a chemical mediator to enhance or boost the immune response along with the protein. But how is it a little bit different? Well, first things first is that we, along with others, and rooted in the work that myself and my co-inventor, Dr. Gordon Joyce, had done at the Vaccine Research Center, which was responsible for engineering the version of the vaccine that is in all the different platforms that are used in the U.S., we take a structure-based approach, meaning we learn the atomic level topography and geography of the proteins on the surface of these viruses. We get a 3D map, a GPS essentially, of the proteins that elicit immunity. And in the context of SARS-CoV-2, that is the spike protein, that protein that mediates attachment of the virus and fusion of the virus with your cells, particularly your lung cells. And then we use these maps to design vaccines and therapeutics, but it's an iterative cycle. So we sort of modify and optimize the engineering of these proteins and see how they do in animals, and if they don't do great, then we optimize it some more. So that was really a major part of the development of these vaccines. But all this happened years before the pandemic occurred, so that when the pandemic emerged, we were ready in many ways. So we think the structures matter, and a lot of this work was done in the context of another respiratory virus, respiratory syncytial virus, but also on the top middle here you see that little soccer ball like image. That is the platform in which we deliver the structurally modified antigen. And you can deliver these antigens in different ways. mRNA, virus vectors, and we're taking the approach of a nanoparticle, a scaffold, essentially, on which we put the spike protein. And in this case, we're using ferritin. Let me get into what exactly ferritin is. So I'll talk a few minutes about the design of our vaccine. We knew from very early on, really from the first day, that we wanted to take a long-term, broad approach to this coronavirus, as we had been trying to do that in the context of other virus families as well before the pandemic. So January of 2020, we started taking an approach to develop a more universal approach to coronavirus vaccines, and in January 2022, Dr. Fauci and others really made the case that we do, in fact, need more universal approaches to coronaviruses. And this was really in the wake of Omicron emerging. And at a White House briefing on the same day, he highlighted several different promising approaches, one of which was ours in the right upper hand corner. And the idea is that, and they're all nanoparticle approaches. Why is a nanoparticle a potentially good option? Well, you can put different antigens, different spikes in this case, or different parts of the spike on a single particle. That single particle then interacts with a B cell. B cells are the cells that generate antibodies. Well, if a single B cell is interacting with two different spikes at the same time, first of all, it's giving it a boost in terms of the signals to produce antibodies, but also what it's going to do is it's going to focus on those common areas between the two different antigens and develop antibodies against conserved parts of those antigens, and so that you get antibodies that are more broadly active. So there are different nanoparticles that you can use. We chose ferritin as ours for several reasons. Ferritin is ubiquitous across life, in animals, plants, bacteria. And so we use a ferritin from a bacterium, H. pylori, which can cause gastric ulcers, because we wanted to choose something that was very far from human ferritin, because if you develop any antibodies against the ferritin part, that could be problematic if it's developing antibodies against human ferritin. Ferritin is very important in carrying iron, which is important in your synthesis of blood and carrying oxygen around. So we don't want people to develop antibodies against ferritin where it could cause a severe anemia. So we chose ferritin, one, because it's ubiquitous, readily available, and we have options to use versions that are very far from human. Two is that ferritin has some interesting geometry. When you express ferritin, it actually polymerizes on its own into this 24 component cage. And when all these 24 ferritins come together in this polymer cage, they form this symmetry, and it's a threefold symmetry at these different juncture points. That's important because proteins like the spike protein and many other proteins that are important for fusion of viruses with human cells have a threefold symmetry. So the spike protein in nature on viruses is not just a single spike protein, it's a triplet. It's like a little bouquet of 3 spikes wrapped around each other. So if we want to present these spike proteins in the conformation that they occur in nature on the surface of viruses, we need something that will present it in that triplet form. And you need that threefold symmetry, and that's what ferritin has. Another reason why we chose ferritin is because we had worked with it at the Vaccine Research Center at the NIH in the context of the universal flu vaccine program. And this particle for flu had already gone into clinical trials. And so we had some safety data for ferritin that we could cross-reference when we took our vaccine for COVID into clinical trials. And you essentially can link whatever you want. You have to do some modifications in engineering, but you can link whatever you want to that ferritin. In this case, we use either the spike or some component of the spike, and it decorates on its own, assembles, and then now you have decorating the surface of the scaffold ferritin all around your antigen of choice. So we went through a extensive but accelerated design cycle in which we developed 215 different candidates in the course of 2 months and tested them in about 2,000 mice. And we came up with 4 different categories of ferritin nanoparticles. Up on the left-hand corner is a structural model of the spike ferritin nanoparticle. Down at the bottom is a receptor binding domain ferritin nanoparticle, and then the 2 right-hand ones are versions of that contain receptor binding domain, but are a little bit larger, the S1 subunit, but are not the entire spike protein. Ultimately, through all the mouse experiments we did, we found that the spike ferritin nanoparticle was really the best option, best version. And we used the pre-fusion stabilized version that was developed at the NIH and is used in all the vaccines in use in the United States. So we went through a series of optimization cycles. We had to make modifications to the base of that spike protein, the hinge regions, in order to ensure stable and natural presentation of the spike protein on the surface of the ferritin. And then we coupled this with an adjuvant, that chemical booster I was talking about, that had been developed in the context of the HIV program at our institute, called ALF-Q, Army liposomal formulation with a SpFN, QS21. That is in at least one other vaccine, in the Novavax vaccine, at least a version of that. Not ALF-Q in particular. ALF-Q is only being used in our vaccine. And here is an electron microscopy image that shows the particle, and so we call it SpFN, spike ferritin nanoparticle. So SpFN nanoparticle along with these globular images that represent the adjuvant. So I'll go through some of the preclinical data pretty quickly here because I want to have time for questions, but this can all be summarized at least initially in this slide, in which we tested our vaccine in mice, in hamsters, in monkeys, and in honestly, actually, in additional species as well, in horses, in sharks. I can answer questions about why we did those other species too, but the pattern that kept recurring was that this was a very potent vaccine. It elicited very potent neutralizing antibodies and very broadly neutralizing antibodies against multiple variants of SARS-CoV-2 and even other SARS-like viruses. And so from our mouse studies, we saw neutralizing antibody titers that were on the level of 10,000 to 100,000 inhibitory dose at 50%. When we took antibodies and purified them from the mice that were vaccinated and then transferred them into other mice, K18 mice, transgenic mice, and then challenged them with SARS-2, we saw those top green and red lines are superimposed. These animals were completely protected. They all survived. And then as we brought down the dose of the antibodies, predictably the survival went down as well. We then tested our vaccine in hamsters, and we tested it against the alpha and beta strains of SARS-CoV-2 after 2 immunizations. Again, like the mice, we saw very high antibody responses, although these are all binding data, not neutralizing data. What we saw was that those animals, those hamsters, and hamsters are a good model. We found, the whole field found, in the context of SARS-CoV-2, they show considerable weight loss and lung pathology, at least for all the variants previous to Omicron. But whether they were challenged with alpha or beta, those animals that were vaccinated either with a high dose or even a low dose of the vaccine twice were very well protected compared to those that were not vaccinated. Essentially, no virus or very little virus found in the lungs of vaccinated animals, or very little virus in the upper airways, the nasal turbinates of these animals that were vaccinated. We looked at 2 different species of monkeys. This represents just one of those experiments. And what we found in the field over the past couple of years is that what you see in monkeys is not always the case for emerging viruses, but in this case, what you see in monkeys is predictive of what you would see in humans in terms of vaccine efficacy. And we saw, again, very high neutralizing antibody responses against the wild-type version, the original Wuhan 1 strain. We saw very strong Th1 or CD4 T-cell responses and engaged memory responses. So we were triggering, at least from the outset, memory T-cell responses and B-cell responses. And we were seeing very rapid protection against challenge in either the lower airways from bronchoalveolar lavage on the left part of the slide, or in the upper airways from nasopharyngeal swabs on the right side. What was also unique about our study is that we had a very potent stock of virus that we were challenging with. You can see in the left of each graph in the black, that's the animals that were not vaccinated. So those are the controls to see what would happen if you weren't vaccinated what were the levels of virus in those animals, and they got very, very high in the upper and lower airways. In the upper airways, around 10 million copies per milliliter, which is much higher than all the other studies that had been done with other vaccines to date. And despite that very rigorous challenge, with 50 micrograms, 2 doses, we saw a very rapid, almost sterilizing immunity. And this bore out in the pathology as well. If animals were vaccinated with 50 micrograms of SpFN twice, essentially were protected from any lung pathology as well. And there was really no presence of virus in the lungs of vaccinated animals as compared to the controls as measured by immunohistochemistry. So the other thing we wanted to look at was the breadth of response. And so we started out looking at the response of elicited by the vaccines in terms of neutralizing antibodies against the wild-type virus, Washington 1, and then against alpha, beta, gamma, delta. And we saw that there wasn't much of a drop in antibody response when we looked across the different variants. And here, in this slide, you can see whether we look by authentic virus in these monkeys who got either 50 or 5 micrograms of our vaccine twice or 50 micrograms once, or looking by pseudovirus neutralizing assays. One, you'll see that there's just very high neutralizing antibody responses to begin with. But if you look all the way to the right in that ID50 GMT fold change, what we're looking at is the fold reduction or increase in neutralizing antibody response against the variants compared to the original Washington 1 strain. And if you look just at the top slide here, the top row, 50 micrograms, 2 doses by authentic virus, you see actually a better response against the alpha, but then not really much of a reduction in response against beta, gamma, or delta. And actually not even, sorry, I pressed the, here it is. You can see here, not much of a reduction in response against those variants compared to the wild type. So you're getting just as good a response as the variants as you are against the original strain. And we did a separate experiment separating our vaccine first dose and second dose by 2 months rather than 1 month. And we challenged with Omicron, and we saw very good responses against Omicron as well, which is not being seen with a lot of other vaccines. And so we saw responses with that 50 micrograms that were nearing, it was close to 10,000 again. And we also saw that there was a response against other sarbecoviruses. So at the top here, you see these different colored bubbles. This is the different clades of on the phylogenetic tree of the sarbecoviruses, the SARS-like viruses. And SARS-2 is in that light blue clade 1b. SARS-1 is in clade 1a. And we're getting very good responses against both of those, but we're also getting responses against clade 3 and clade 2. These are binding antibody responses by something called biolayer interferometry, or we just call it octet by shorthand. And this has been very predictive of neutralizing antibody responses as well. We're developing those viruses now for or testing against some of those viruses in neutralizing assays. But just by binding, we're seeing a good response as well against a broader panel of sarbecoviruses. And when we challenge with Omicron from that monkey study I was referring to, again, we see very good neutralizing antibody responses after 2 doses. just before challenge, so week 12. And then when we challenge with Omicron, this is the BA1 subvariant of Omicron, and not BA2 or any of the others, we see a very rapid protection in the animals that got SpFN compared to those that didn't get vaccinated in either the lower airways in the top here, BAL, or in the nasal swabs in the bottom. So we are now completing our first phase 1 clinical trial with our vaccine, and the design is dose escalating. We vaccinated individuals either with 25 micrograms or 50 micrograms of our vaccine at 0 and 1 month. And then there was the option of getting a 6-month vaccination as well. A subset of individuals did get a 6-month vaccination. The second group was a 50 microgram arm. And unfortunately, we were trying to get the trial started quickly, but it didn't start until April 6th of 2021. So people had started getting vaccinated, and we were enrolling individuals who were naive to vaccination or infection. So it became difficult to enroll pretty quickly as vaccinations rolled out. But we still got enough participants to assess safety and immunogenicity, or tolerability and immunogenicity, and now are analyzing those data, and they look very promising in recapitulating what we saw in the animals, and hopefully I will be able to present those data publicly in the next few weeks. And what I'll be presenting is pseudovirus neutralizing antibody titers, not only against the original strain, but all the variants of concern through day 209, so all the way through 7 months after initial vaccination, binding antibodies, cell-mediated immunity, and then looking at breadth as well. So broader sarbecovirus, pseudovirus neutralizing antibodies, broader sarbecovirus binding antibodies. And then we also have a experiment that we're doing in which we're taking antibodies, purified IgG, from vaccinated individuals and then transferring them into hamsters, and then challenging those hamsters either with Omicron or SARS-1 to see if the antibodies elicited by our vaccine are protective. It's a sort of a surrogate way of assessing efficacy, since we can't really do that in the context of a clinical trial, a phase 1 clinical trial. So what's next? We're going to be looking at nanoparticle combinations. We've already started doing that, combining spike ferritin nanoparticles from SARS-2 with those for SARS-1 or other coronaviruses, which we've started making those as well. We've already seen that cocktails of these vaccines elicit very potent neutralizing antibody response against multiple SARS-like viruses. Developing more mosaic antigens, so not just not cocktails, but the single particles that present multiple spikes on the same particle. And then looking at this with adjuvants or not with adjuvants, and looking with different platforms, looking at expressing our construct, our sequence for nanoparticle from mRNA. So marrying the benefits of mRNA platforms with nanoparticle platforms. So I hope that I've given you a little bit of a flavor into the general approach and our specific approach towards developing broadly active platforms against coronaviruses that can then be translated to other virus families. And that now we're getting into a phase of this pandemic that hopefully is becoming more of an endemic disease, and now we can start preparing for future threats. And I'll leave you with a quote, since I started you with a quote, and this is a little bit philosophical from the Danish philosopher Søren Kierkegaard, in which he said, "Life can only be understood backwards, but it must be lived forwards." And the meaning of that in the context of the pandemic is that, yes, we have to look back, be introspective, retrospective, try to understand what we could have done better, but we have to apply that going forward. I think a lot of times when things fade, we tend to forget, we tend to divest, and now is the time where we need to invest more than ever in these kinds of platforms for the future, so that the next time something emerges, a potential pandemic, we actually have the vaccines on the shelves that have already been tested in humans that we can start deploying right away and don't even have to wait a year to save millions of lives. Thanks for your attention, and I'm happy to take questions.
And thank you, Dr. Modjarrad, for that informative presentation. We will now start the live Q&A portion of this webinar, and if you have any questions you want to ask, do so now, but just click on that Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have a great set of questions coming in. Our first question, as compared to the whole pathogen vaccine platform, a protein subunit vaccine, what are its potentials for a global COVID vaccine?
Thanks for that question. So I'm assuming that the reference to the whole pathogen vaccine platform are the whole inactivated virus vaccines, which have not shown to be as effective either against the wild type or against variants of concern, and largely because we think the spike protein on the surface of those inactivated viruses are in this post-fusion state. And the post-fusion epitopes that are presented aren't as immunogenic as the pre-fusion stabilized version. We saw this in the context of RSV as well, and now we've seen it in the context of coronaviruses too. So we are taking the approach of protein subunits. So either the spike protein, which is the whole protein, or a subunit, either the S1, or really where a lot of the focus has been on the receptor binding domain, that very specific part of the spike protein that interacts with the human ACE2 receptor. The benefit of RBDs as well is that they don't have to be presented in that triplet or trimeric conformation. They can be monomers, and that gives you a lot more flexibility when it comes to developing mosaic particles, where you can have RBDs from multiple different viruses and you can have more of them on a single particle. And so that definitely is one of the approaches that we're taking and other groups are taking as well.
Thank you so much. Our next question, with ferritin nanoparticle platforms, manufacturing challenges related to particle heterogeneity, improper folding of fused antigens, and antigen interference with intersubunit interactions still need to be overcome. What are your thoughts on that?
Yeah, manufacturing, so the CMC issues are an important aspect of this, and I appreciate that question. Something that is being thought through on a global level, within the federal government, by the WHO, by organizations like CEPI, the Coalition for Epidemic Preparedness Innovation. Some of this has, we're also kind of borrowing from the work that's been done in the context of the flu program as well, and interactions with the FDA to develop validation processes that give an indication of how much of each antigen is present and is being presented in your vaccine formulation, in your drug product, and ensuring that you can predictably and consistently quantify that from lot to lot as you manufacture this product. And on a small scale, it's worked very well. We've been able to do that. We've been able to demonstrate antigenic integrity, nanoparticle integrity, consistency. Every nanoparticle pretty much is, presenting the same thing, same antigens in the same ordered array. But when we get to scaling, that's a big issue. Other groups, there's one other group that has had some of this issue in terms of scaling. So we'll have to work on those processes as we try to move from making thousands of doses to billions of doses. And that was an issue also for even vaccines for other platforms in the context of this pandemic as well, not just the nanoparticles.
Thank you so much, Dr. Modjarrad. And I want to thank our audience for these great questions coming in. This is awesome. Our next question, a major challenge of ferritin-based vaccines will likely be related to antigen design. Example, whether its structure can be maintained and or it's capable of inducing immune response after conjunction to the ferritin. Any advice about this?
Yeah, so we found that at least for flu, first of all, for coronaviruses, and we've been doing work a lot on arenaviruses as well with the GPC protein, that we actually get very consistent presentation of the native conformation of these class 1 fusion proteins on ferritin. Ferritin is a much smaller nanoparticle than other nanoparticles out there, which have some benefits and drawbacks. So I think there was some the previous question asked about sort of maybe some interference. If the sort of the business and the most important parts immunologically is at the head of the protein that we want to induce a response against, like the RBD, then it's not so much of a problem with ferritin and its size. But if it's sort of getting towards some of the sides, or if you want to get better responses against sort of the stem, or in the context of coronaviruses, the S2 subunit, then it becomes a little bit more difficult for the B cell receptors to access some of those epitopes in the context of ferritin, as opposed to some other nanoparticles that are being developed. But in terms of the structures of the proteins that have been expressed on the surfaces of these ferritin nanoparticles, we've seen very good expression in the conformation that we wanted to be presented in so far. It does take some additional, so it's not just a matter of taking S2P or RBD and just plopping it on. There is additional design that has to go into this. I showed for a spike ferritin, we had to make some modifications to the hinge region of what was already modified with the S2P. And then with RBD, we not only had to kind of play around with the part of the RBD that was proximal to the 6 amino acid linker, we also had to modify our ferritin. So our RBD ferritin nanoparticle is actually a hybrid or a chimera of frog ferritin and H. pylori ferritin, because with H. pylori ferritin, we just couldn't get the RBD presented in a full nanoparticle to form. So there is still quite a bit of playing around with the design. That's why we need to do these things now before the pandemics occur, because every day in the context of the pandemic is very, very precious, whereas if we're in an inter-pandemic period, we have a little bit more flexibility in taking a little bit longer in optimizing the design.
Thank you so much. One of the most prominent features of mRNA vaccine is no real memory and no adaptive evolution. Isn't it time to say that mRNA vaccine isn't qualified to be a vaccine? And the second part of that maybe is just that it should be another class of antibody-based drugs, but not actually called a vaccine.
Well, there's still debate about that in terms of how much memory mRNA vaccines induce, because we do see that there is both T cell and B cell memory. It may not protect so well against, and remember, right now, so all these mRNA vaccines are based on the Wuhan 1 or Washington 1 strain, and we're looking at the efficacy against Omicron, which is, almost like is very close to being a different species of coronavirus. And so one issue is sort of the breadth of response that you're getting. And then the second issue, yes, is the memory, but there is definitely memory in terms of the protection against death and severe disease. Against infection alone, it's not only necessarily systemic memory response, but the issue that is problematic for all of the vaccines is the mucosal response as well, to prevent replication, particularly with Omicron in the upper airways. So I think that's problematic for all the vaccines. And mRNA vaccines are in their adolescence. They're no longer in their infancy, but they're not fully mature platform. This is the first time that they've hit the prime time, and so there's still quite a bit of optimization that has to take place. There are definitely some disadvantages to the mRNA platform. It is not a panacea, but there are some clear advantages in terms of scalability, first and foremost, for mRNA platforms. But they do need to be optimized better, not only the mRNAs, but the lipid nanoparticle formulations in which they come, in which they're packaged, to elicit broader and more durable responses.
Thank you so much, Dr. Modjarrad, and I want to thank our audience. Do you want to provide any closing remarks before we go?
Well, first and foremost, thanks again to you, Susie, and to the organizers and to LabRoots for giving me this forum and this opportunity to engage with the audience. Thanks to everybody for their questions. I'm happy to have follow-on discussions afterwards. The organizers have my email. And I hope this was informative and gives you some insights into where the field is headed. We're not the only group doing this, but we're part of a small group and growing group that has been looking at broadly protective approaches to emerging viruses in general, not just coronaviruses, and how we need to do business differently. We don't need to come back to normal. We need to come back to a new normal and how we anticipate and prepare for future pandemics.
Absolutely. And Dr. Modjarrad, thank you again for your time and for your important research. And before we go, I want to thank our audience and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years up to May 4th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay safe, everyone. Take care. Bye-bye.
Thank you.